All right, good morning, everybody. Let's get started. So I think we finished like glycolysis, right? I mean, we're talking about, we'll talk about the under different conditions, aerobic, anaerobic conditions, the glycolysis final product can be different, and also the ATP production, the amount of ATP produced from different pathway can be also different, right? And this, occurred in a different type of cells, and we give a sample like red blood cell, right, and versus the oxidative cells, and then basically the under an anaerobic condition, the final product of glycolysis is lactate, right? And the aerobic condition product is pyruvate, and pyruvate can be transported into the mitochondria for further oxidation, and generate final product like CO2 and water, and of course produce a lot of ATPs in the TC cycle in mitochondria, right? So remember this process. So now we're going to move on to the TC cycle, the next step under the aerobic conditions. So mitochondria, this is mitochondria. I guess you guys learned, right, from other courses, and mitochondria has actually two membranes, double membrane, the inner mitochondrial membrane and outer mitochondrial membrane. So in between there is a kind of space, right? And also a mitochondrial matrix. So lots of enzymes, particularly the enzyme that involved in the respiratory chain or oxidative phosphorylation, located in the mitochondria. So that's why mitochondria is so important, because electron transport chain, right, is occur in the mitochondria. So oxidative phosphorylation can happen in the mitochondria. So finally, when we generated reducing equivalent like NADH or FADH from either from mitochondria or TC cycle, for example, right, in mitochondria, or from the glycolysis in the cytosol, eventually will have to go into the mitochondria for oxidative phosphorylation for ATP production. So an important enzymatic complex is called a pyruvate dehydrogenase complex. Remember, pyruvate, if it's generated from glucose glycolysis, glucose oxidation in the cytosol, and transported into the mitochondria. And the first an important reaction is pyruvate has to be converted to acetyl-CoA, right? So, and this reactions, the pyruvate converts to acetyl-CoA, occur in the mitochondria and catalyzed by this complex, pyruvate dehydrogenase complex, right? So complex means this enzyme consists of several multiple enzymes that here actually the three enzymes, right, they work together to catalyze this whole reactions. So I just want you to remember this, the prosthetic group, some like vitamins, that actually involved in these enzymatic activities, right? So yeah, just give you idea. This is a complex. Three enzymes form this complex and also involved in the prosthetic group that is important for enzymatic activities. And the regulation of pyruvate dehydrogenase is also important, right? The activity is not always active, depend on the energy needs. So the two important substrates can regulate the enzyme activities. One is the acetyl-CoA, which is a product, right, of this enzymatic reaction, and the other one is the NADH. So NADH is also generated from this reaction. So acetyl-CoA and NADH can serve as sort of negative regulators, right? Can inhibit the pyruvate dehydrogenase activities. So, for example, that probably makes sense, right? If we have already made a lot of energy, NADH or acetyl-CoA molecules generated, then we need to tell the enzyme to slow down. We don't need to actual energy, right? So these two products of the enzymatic reactions go back to inhibit the enzyme activity to allow the reaction to slow down. And the other thing important I want to tell you is that the acetyl-CoA not only be generated from glucose, from glycolysis glucose metabolism, it can also be produced from amino acids and fatty acid oxidation, right? So acetyl-CoA, again, we're going to talk a lot later, is key, a very important, essential molecules in the energy metabolism. So kind of connect the three macronutrients metabolism, oxidative metabolism, right? Again, so acetyl-CoA can be generated from glucose, oxidation of glucose, amino acids, and fatty acids, right? All right. So acetyl-CoA, and I just talk about it, right? It's again the central molecules and produced from glucose or glycolysis or amino acids metabolism, oxidation, or fatty acid oxidation, right? And acetyl-CoA is also the key molecule that enter the TC cycle, right? Enter the TC cycles for a complete oxidation if we need energy for producing ATPs, and if under the if we have too much energy and we don't need that much energy, so we can also convert it the glucose or amino acids to fatty acids, right? So acetyl-CoA can also be used for generation of fatty acids synthesis, ketone body synthesis, and the cholesterol synthesis, right? So again, it depends on the physiological conditions. For example, during the fasting, acetyl-CoA will not enter the TC cycle for complete oxidation, but will be used for ketone body synthesis, right? We will get there later. And under the fat condition, when we have a lot of acetyl-CoA generated from glucose, amino acids, fatty acids, right? We eat not just the glucose, right? I mean, carbohydrate we also have. In our diet also have a protein and lipids, right? So eventually we will produce a lot of acetyl-CoA during the fat condition. And this acetyl-CoA can be entered the TC cycle for complete oxidation for produce ATP, but also to generate the citrate. So citrate is very important that can leave the TC cycle and enter the fatty acid synthesis pathway, right? So you can convert it surplus energy either from amino acids or from glucose to the fat, right? So this is sort of the pathway how fat is synthesized in the fat condition when we consume extra energy, right? All right. So just keep in mind this is a very important central molecule that connect all metabolic pathways, right? Yes.
I'm confused. Could you repeat that part? In fat condition, it can be used to generate citrate and also participate in the Krebs cycle. And what else was it?
Oh yes. So in the fat condition, right? When we have a lot of acetyl-CoA produced, and then it can enter the. It can have a two pathway, right? One pathway is enter the TC cycle, a complete TC cycle, right? And for ATP production, which we're going to talk about just the next couple of slides. And the second pathway, extra. We don't need extra energy, right? So an extra acetyl-CoA can also be converted to citrate, and citrate that leaves the TC cycle and go to for the fatty acid synthesis, right? So next slide, I think it will show you, give you a better idea, the more clear. Okay, now let's talk about the TC cycles. TC cycle is very important. If you remember, it's so-called amphibolic metabolism, amphibolic pathway that can link between an anabolic and catabolic pathways, right? So kind of connected, or synthetic pathway or catabolic pathway. It's a common pathway, like I already mentioned, right? It's a common pathway for the 3 macronutrients metabolism. So carbohydrate, protein, lipids can be metabolized and generate the acetyl-CoA, right? So acetyl-CoA can be produced from, again, metabolism of 3 macronutrients. And this can enter the TC cycle, right? Acetyl-CoA enter the TC cycle either for complete oxidation, so during the TC cycles, a lot of ATP produced, either directly from the substrate levels phosphorylation or produce NAD, FADH, and then this FADH and NADH and FADH will enter the oxidative phosphorylation, your electron transport chain, right, for converting, conversion the ATP, energy to ATP, right? So TC cycle is very important. Okay, so this is the whole TC cycle reactions. So again, acetyl-CoA is the first molecules, right, that enter the TC cycles. And you see the first reaction, the product is a citrate, right? So acetyl-CoA can be converted to a citrate together with oxaloacetate. So citrate, again, can leave the TC cycles and go to fatty acid synthesis, so lipogenesis pathway, right? And it also can continue, and go through the TC cycles for producing ATPs. So you can see, but you don't need to remember all the, detailed reactions, right? Again, you just try to know, the different reactions and something, but you don't need to memorize each reaction, right? And ATP production, you try to remember which steps that produce ATPs and how many ATPs, so NADH, FADH is produced into that reaction. So I just circled, the reaction that produce either direct ATP or the NADH, right? So you can see that one ATP is produced in this reaction, right, at the substrate phosphorylation level, and 1, 2, 3, 3 NADH molecules are produced per 1 TC cycles, right, and 1 FADH is produced. So again, if acetyl-CoA molecules go through the whole complete TC cycles, will generate 1 ATP, 1 FADH, and 3 NADH, right? So, and then this NADH or FADH will eventually go again to the phosphorylation, right? Oxidative phosphorylation or electron transport chain for ATP production. And just wanted to remind you that the how many acetyl-CoA molecules are produced from one glucose? Two, yes, thank you. Two, acetyl-CoA molecules produced, right, from oxidation of one glucose molecules. So when you calculate ATP, to keep in mind, you need to double, right, the ATP per 1 TC cycles. And the other thing I wanted to mention that the TC cycle is very important to connect the all three macronutrients metabolism, right? The fatty acid synthesis I already mentioned. This is very important. Try to remember this. This is a pathway that convert glucose to fatty acids, right? If we have too much glucose, is consumed, and also amino acids can also enter the TC cycles, right? For oxidation and also for converted glucose. So we're going to talk about later, right? So this cycles is very important. The TC cycle also participate in the gluconeogenesis, right? And when amino acids used as a precursors, so part of TC cycles, is actually is part of gluconeogenesis. All right. Any questions? Yes.
What do the amino acids do in a cycle? I mean, what is what are they doing when they enter? Do they enter to add to the reaction to keep it running, or do they or process, or what happens to them?
That's a good question. So we're going to talk about the anapyrolysis and catalysis, this process. So that means that the amino acids, for example, during the fasting, some of the amino acids can be used as a precursor for gluconeogenesis, right? So converted non-sugar precursor to glucose. And so, for example, here, actually glutamate can be converted alpha ketoglutarate, right? And then through all this reaction to generate the oxaloacetate. So oxaloacetate is one of the key molecules in the gluconeogenic pathway, right? So and then oxaloacetate can leave the TC cycles and continue on the gluconeogenic pathway and for glucose production. So this, so amino acid can go through part of the TC cycle, right, reaction and for gluconeogenesis. Okay, so calculation of ATP production. We already know, right, under the aerobic condition, 6 to 8 ATPs are produced from glycolysis, right? Remember, from glucose to pyruvate. And then if cells at the mitochondria and under the aerobic condition, so the pyruvate then enter the TC cycle, enter the mitochondria, and the first reaction would be the pyruvate converted to acetyl-CoA, right? And this is a reaction catalyzed by pyruvate dehydrogenase complex. Remember we mentioned, right? And this reaction actually produces produces one NADH molecule. Remember, right? And because this NADH molecule is already inside of mitochondria, so you don't need a shuttle to transport. So this this one NADH molecules can produce 3 ATPs, and because we have 2 pyruvate molecules are produced from one glucose molecules, right? So times 2, 3 times 2. So you, in this particular reaction, you will generate 6 ATPs. And in the TCA cycles, one TCA cycles, we produce how many ATPs? Do you remember? You can think about how many ATPs can be produced from one from the TC cycles. One TC cycle. One is from phosphorylation, right? Substrate level phosphorylation, and 1 FADH, 3 NADH. So calculate how many ATPs total per one TC cycle. How many ATPs produced? 12, yes. 12. So one TCA cycle is 12, and we have 2 acetyl-CoA enter the TCA cycles, right? So times 2, 24. So the total, if one glucose molecule is completely oxidized to CO2 and water, will generate 36 to 38 ATPs, right? So I think this is a good exercise, just allow you to go through the whole metabolic pathways, started from glycolysis and pyruvate dehydrogenase reactions and TC cycles, and to the whole process for the complete oxidation of glucose, right? So this is a good exercise, when you calculate ATP production. Okay, so any questions about glucose glycolysis and TC cycle that we just finished? No? Okay. All right, let's okay, so let's move on. The second important metabolic pathway is glucose in gluconeogenesis, right? So gluconeogenesis, this just goes through again, glycolysis pathway, right? Just refresh your memory that one 6-carbon glucose molecules can be oxidized through the glycolytic pathway in the cytosol, produce 2 pyruvate molecules, and then a pyruvate, if under the anaerobic conditions, are converted to lactate. But if under the aerobic condition, they can be further oxidized inside of mitochondria, right? And pyruvate is converted to acetyl-CoA through the pyruvate dehydrogenase complex reactions, and acetyl-CoA is the first molecule that enters TC cycles, right, for complete oxidation. And this is something that energy that produce 2 ATPs, 6 NADH, and 2 FADH, and this reducing equivalent enter oxidative phosphorylation and produce ATPs, right? 36 to 38 ATPs produced eventually in the mitochondria. And now let's talk about the respiratory chain or oxidative phosphorylation. Remember the glycolysis and TC cycle generally lots of NADH and FADH, and this reducing equivalent contains energies, right? But you need a chemical reactions to convert the energy from NADH, from reducing equivalent molecules to the ATPs. So, and electron transport chain is a place that converting energy from reducing equivalent to ATPs. And electron transport chain, it consists of 4 complexes, right? So you still remember complex 1, 2, 3, and 4. So, and these 4 different complex actually contains a different enzymes, right? So, multiple enzymes has formed the different complex. And what you need to remember is the complex 1, 3, and 4 are the proton pumps, right? 1, 3, 4, complex 1, 3, and 4 are proton pumps, meaning that this enzymatic localized in the inner mitochondrial membrane and can function as a pump, proton pump. So can pump a proton out of inner mitochondria cross the inner mitochondrial membrane and generated a proton gradient. So this proton gradient contains energy, right? So so that means only this 1, 3, and 4 complex can generate proton gradient and can produce ATPs. The energy can be used to produce ATPs. And complex 2 is basically localized inside of the mitochondrial matrix. It's not on the membrane. So complex 2 is not able to generate proton gradient, not able to generate energy, right? So again, only 3 be able to generate energy because of the proton pump. And also the inner mitochondrial membrane has the ATP synthase. ATP synthase is the ATP dependent proton pump that is a place that eventually produce ATPs. So the enzyme the reason we call this oxidative phosphorylation, that means that the whole electron transport chain contains the so-called oxidation and reduction reaction series, right? So many oxidation and reduction reactions happen that can transport electron from reducing equivalent to the complex 1, 3, and 2, and eventually convert it to energy into ATPs, right? So here I think it gives you a better idea, more clear. You can see that again, complex 1, 3, and 4 are the proton pump and located on the inner mitochondrial membrane, right? They can pump protons out of mitochondria and cross the in the mitochondrial membrane and generating proton gradient. And this proton gradient eventually be allowed proton transport back to the matrix and by the enzyme called ATP synthesis. So the ATP synthesis is also the proton pump, but they pump proton from the opposite direction, right? From the outside the mitochondrial matrix and to inside the mitochondrial matrix. So through the direction, that energy can be transferred to ADP and to form ATP, right? And complex 2 again is localized in the cytosol, right? You were not able to generate the proton gradient. So if the reducing equilibrium molecule transport electron to the complex 2 will not generate the energy, right? So here maybe give us a kind of explanation why NADH can produce 3 ATPs while FADH only produces 2 ATPs, right? Because NADH will transport electron to complex 1, right? And then to complex 3 and 4. So can generate the 3 different proton gradients, right? So that's why NADH eventually can produce the energy, the contained energy that enough to produce the 3 ATPs, right? And FADH, they usually transfer its electron to complex 2 and then 3 and 4. So that's why the reason only produce the 2 different electron gradients, right? Only enough for 2 ATP production, right? Does that make sense? Okay. All right. Remember we mentioned that NADH that produced through the glycolysis pathway in the cytosol can generate either 2 or 3 ATPs, right? Remember? So the reason here, we're going to explain the reason why sometimes NADH from glycolysis in the cytosol only produces 2, sometimes can produce 3. Because NADH has to be transported into mitochondria then for oxidative phosphorylation, right? For ATP production. But NADH is not able to pass through mitochondrial membrane. So it needs some help, needs some shuttle or transporter to help transport NADH to the mitochondria for oxidative phosphorylation. So there are actually 2 shuttle systems. One is called a glycerol 3-phosphate shuttle, and the other one we're going to talk about next slide called a malate aspartate shuttle. So 2 different shuttles can transport the electron from NADH that's generated in the cytosol into the NADH or FADH in the mitochondria. So let's talk about the glycerol 3-phosphate shuttle first. So NADH, for example, is generated in the cytosol from glycolysis, right? Can transfer its electron to dihydroxyacetone phosphate and to generate a glycerol 3-phosphate. So glycerol 3-phosphate carried the electron originally from the NADH, right? And glycerol 3-phosphate can pass the electron through the outer mitochondrial membrane to the to the FAD in the mitochondria, right? So it is the FADH that actually eventually enters the electron transport chain for ATP production, not the NADH. So NADH in the cytosol is NADH, but when it's the electron is transported to the FADH, and in the mitochondria it becomes FADH, right? Does that make sense? So that's why they only produce the 2 ATPs. Does that make sense? So because of some type of cells, they only express glycerol 3-phosphate. They only have a glycerol phosphate shuttle. So in some type of cells, and the NAD electron in the NADH in the cytosol, we pass through the FAD in the mitochondria because they only have a glycerol 3-phosphate shuttle, some type of cells, and do not have a malate aspartate shuttle. So that's why in some type of cells, the FAD produced in the glycolysis only produces 2 ATPs eventually. Right? That makes sense? Right? And this is a malate aspartate shuttle. So malate aspartate shuttle is probably present in the other type of cells, right? So the NADH again itself cannot directly pass through the inner mitochondria, right? So it has to be converted to the had to be transported its electron to oxoacetate and generating malate. So malate is able to penetrate inner mitochondria and goes into the mitochondria, and in the mitochondria malate actually pass the electron, which initially from NADH in the cytosol, right, and to the NAD and convert it back to NADH in the mitochondria. So this NADH in the mitochondria that eventually enter the electron transport chain for producing 3 ATPs, right? So that's why sometimes when in some type of cells, if they have a malate aspartate shuttle, NADH produced in the cytosol can produce 3 ATPs. Right? Does that make sense? Yes. No, I mean the energy usually they actually lost during this reaction. This right. So they only be able to pass, to electron to the FAD, right, and this somehow is lost during these reactions, right. So so the FAD, because of FADH, is not able to pass the electron to the complex 1, right? Only 2, complex 2. So that's the reason they only produce 2 ATPs, right? That clear? All right. Okay, so now let's talk about the gluconeogenesis, the second glucose metabolic pathway, right? Do we want a break? Okay, yeah, let's take 10 minutes break. Hi.
With questions.
Yes.
For those shuttles, why does it have to happen where the two shuttles that you were explaining, why do those have to happen? Like you said, in some cells it happens,
Right.
but in the general sense of it, it doesn't happen regularly. So like you said, the NADH gets transferred through into the inner membrane,
Right.
and it's giving the electrons to the FAD.
Right.
Now, why is that happening?
Oh, because of NADH is not able to go through the membrane, go through the inner membrane. Inner membrane is not permeable to NADH molecule.
And this is in the cytosol. It's made in the cytosol, the
Right,
NADH.
right,
and
right.
when is this happening in the process of everything then? So is the NADH coming from like early on in glycolysis then?
Right.
Not in the TCA.
Not in tissue.
This is from early
This is from the
glycolysis.
glycolysis. Right.
Cytosol.
Right.
And that's when that's where the FADH2 comes from. That goes into electron transport chains.
That's true. That's true. Because for making ATPs, you have to go to the electron transport protein, right? Which is the mitochondria.
So you're just saying the NADH here just can't pass through that. So
Right.
But then the only way it
Right.
can be transferred over the electrons can only be transferred to FADH2, not another NAD or anything. There
No,
wouldn't be that.
no. With this shuttle.
With the glycerol.
Yeah, we only.
And the second one, what's
And the second one,
So it looks
that
like
is,
they're transferred
yeah,
somehow.
you actually, give the electron to the acetate and to make melee, right? And melee is able to transfer,
into the
energy
mitochondria.
in the membrane to mitochondria, and melee give the electron, which initially from this molecule, right? And so this melee is able to give electron to NAD, another different NAD molecule in the mitochondria, so be able to convert it back to the NADH.
So is this just basically recycled? So it's like
Yeah,
oxaloacetate just gets
oxaloacetate.
recycled and keeps going, and
Right.
it has to be alpha gets turned out.
Alpha-ketoglutarate, after we come back, to the.
And that's from glutamate, so that's amino acids that you're talking
Right,
about.
right. So actually, this is more complicated. Involve glutamate, right?
Yeah, you almost just want us to know this top
glutamate.
part mainly. for
Right,
the
right. I
NAD.
don't want to explain too
Yeah.
complicated. So I just thought we just.
This is where we're getting ATP from.
That's true. And
So this is another way of having the same thing happen from the previous slide. So you were saying
right. It's a different shadow. So
Two different
what
shadows.
I try to explain is sometimes, for example, in the brain cells.
Yep.
Basically, most of the shuttle is mainly aspartic shuttle. This is happening mostly in the brain cells, and in the other type of cells, for example, in intestine,
okay.
cells.
Yeah.
And this will
This is
not
more-
happening, and usually more something happening like
this is
in
more
this.
intestinal, you said.
Yeah. So
And then the
to
other one's
depend on the tissue cells.
Okay.
So that's why. So maybe in the brain cells, the NADH produced in the cytosol can produce 3 ATPs, and in the small intestine, cells, and only produce 2.
That's why you
because
say
of,
2 to 3.
Yeah.
Yeah.
So it depends on the type of cells.
Okay. Perfect. Thank you.
Yeah. You're welcome.
It's hard to tell from the diagram though how many are.
Yeah.
Because I just see like here there's one. Here there's one coming out, right?
Yeah.
An ATP. This is where the AT would be coming out. Is that right?
Yeah, the respiratory chain. Yes.
Yeah, but then here, I mean, where is the ATP coming out? Like where?
Oh, ATP. I already explained it here. See here.
Yeah.
And respiratory chain, right? If it's NADH and then it's past this electron proton to the complex 1.
Oh no, I think I understand that part, but just the difference of these two, like.
Oh, okay. So
Yeah.
so this
Sorry.
this is something happen
Right.
or occurred in the mitochondria, right? Because of mitochondria has this all the enzyme, right? This complex actually located in the mitochondrial membrane. Right? So and then remember we also have NADH produced from the glycolysis in the cytosol, right?
Right.
And because this NADH molecule cannot pass through
Right.
mitochondria to finish, the electron transport chain
Yeah.
to get energy ATP.
Yeah.
So that needs quite a shuttle to help, transport electron from NADH to the similar the
Okay.
equivalent
Okay.
right molecules in the mitochondria. So in this shuttle, so the different type of cells have different shuttles. In general, we have two different type of shuttles. Glycerol 3-phosphate shuttle. So the glycerol 3-phosphate shuttle actually pass the electron from NADH, goes through the mitochondrial membrane and in the mitochondria, and then passes electron to FAD molecules in the mitochondria. So this can be converted to FADH, right? And the
Kind of being right here.
right.
Okay.
So you try to connect this here to here. So that's why if cells only have a glycerol 3-phosphate shuttle, and they only NADH produced in the glycolysis, right? And if some type of cells they have mainly aspartate shuttle, and then this will eventually similar, the mechanism, right?
Okay.
So,
Bring it over.
but different, molecules.
Okay.
So they bring over to NADH, not FAD. NADH in the mitochondria. So
So there
they're
is
able
NADH
to
already.
right.
It
so,
did cross the membrane somehow. I
Sorry?
was thinking that you couldn't have NADH in the membrane because it wouldn't be able to cross.
No. I mean
Okay.
the mitochondria had their
Okay.
itself, has the FAD, and you can make FAD inside of mitochondria,
Okay.
right? And cytosol also have their own NAD made. So then this shuttle will be able to pass, the electron.
This is all review, but I just.
Yeah. I know it's complicated. So of course, this shuttle is more complicated and also involved in the mailing. Just recycle the oxaloacetate, back to cytosol, so you can go the next cycle, something like this. So I just don't want it too complicated. I will just try to focus on this.
Okay. And you're gonna be. Do you need a break? I'm sorry. I'm asking
Oh
you
no, that's
questions.
okay.
Are you
Yeah.
sure? So NAD and then NADH. I'm trying to. The different. This is the. When it has the H, it has the electron on it, and when it loses the H, it's right.
Yes,
Okay, so
yes,
this is
right.
the reduced form, and this is the oxidized form.
Yes.
Okay, I just
right.
wanted.
Yeah.
Thank you.
Yeah, you're welcome.
Oh, you can? I didn't know that. You do stop it. Show them.
Technically, they're called also 18.
I mean, they're funny.
I like that goes like this.
Thank you so much.
Okay. All right. We have enough break. All right. So let's talk about the gluconeogenesis, right? This is also important pathway. That is it's a biosynthesis of glucose from carbohydrate precursors, right? And this pathway, the purpose of the gluconeogenesis is to maintain blood glucose within the normal range, right? Gluconeogenesis, the purpose of our body does a gluconeogenesis is to produce a glucose, right, from non-sugar precursors, and we try to maintain the blood glucose levels. So under which physiological conditions do you guys think the gluconeogenesis will happen? Fasting, right? So fasting, when we try to maintain blood glucose level, not allow blood glucose level drop too significantly, right? We need to, make glucose. But of course, the other pathway is important, right? Glycogen breakdown is also important pathway, a similar purpose to maintain blood glucose level. And it occurs, the gluconeogenesis occurs primarily in liver, kidney, tissues, right? We talked about it earlier. And it occurs primarily in which part of cells and intracellular compartments. Say that again. Mitochondria, yes, and we have cytosol. Yeah, cytosol and mitochondria, right? So, and important thing is the gluconeogenesis is converted, non-carbohydrate precursor to glucose, but it is not the exactly, the reversal of a glycolysis. We know the glycolysis we break down glucose, right, to the smaller molecules, and gluconeogenesis we make glucose from smaller molecules to glucose, but the reaction is not exactly the reversal of the glycolysis. And common precursors for gluconeogenesis: pyruvate, lactate glycerol, protonate, and also some glucogenic amino acids. Glucogenic amino acids, that some type of amino acids, if they can be converted to pyruvate, oxaloacetate, or alpha-ketoglutarate, and this type of amino acids can be used for glucose production, for making glucose. So this type of amino acids called glucogenic amino acids, right? For example, alanine, serine, glycine, serine, the cysteine and aspartate, glutamate, right? The others. So you try to remember these precursors, the common precursors for gluconeogenesis, right? Okay, so the reason that why gluconeogenesis is not exactly reversal of glycolysis, remember glycolysis, we ask you to remember the three key reactions, right? 3 key reactions or rate-limiting reactions. And these 3 rate-limiting reactions are irreversible, right? For example, the glucose can be phosphorylated to glucose 6-phosphate by either glucokinase or hexokinase, right? This is one of the key reactions. in the glycolysis, and second reaction is the fructose 6-phosphate can be converted to fructose 1,6-phosphate by the fructose 1 kinase. Right, this is the second key reaction in the glycolysis, and the last one, third reaction, is the pyruvate kinase catalyze the conversion of phosphoenolpyruvate to pyruvate. And the irreversible means that this key enzymes only able to catalyze reaction from right to left. Sorry, from left to right. They cannot catalyze reaction, the backwards, right? For example, glucokinase only catalyze the conversion of glucose to glucose 6-phosphate. They cannot catalyze the conversion of glucose 6-phosphate back to the glucose, right? So that means we need different enzymes to catalyze the reversal reactions. So this 3 reversal reactions catalyzed by a different set of enzymes are the key enzymes in the gluconeogenesis, right? So this is the next slide. So you can see here the key reactions. I just changed the order because from the gluconeogenesis we started from the opposite direction, right? So this would be, for example, if we try to convert pyruvate, phosphoenolpyruvate to pyruvate. If we try to convert it back, pyruvate back to the phosphoenolpyruvate, that requires a different set of enzymes. These 2 enzymes, pyruvate carboxylase and phosphoenolpyruvate carboxykinase, these are the 2 key enzymes that can work together, can convert pyruvate back to phosphoenolpyruvate, right? So these 2 are the key enzyme in the gluconeogenic pathways, right? And same thing for the other 2, the irreversible reactions. And in the gluconeogenesis, the fructose 1,6-biphosphatase are the key enzyme, right? In the gluconeogenesis that converts fructose 1,6-bisphosphate to fructose 6-phosphate. And the last reaction in the gluconeogenic pathways is the conversion of glucose 6-phosphate to free glucose. This is the key enzyme, glucose 6-phosphatase. It's a key enzyme in the gluconeogenesis. Remember? Right? So, and we also mentioned that this is enzyme that also important in the glycogen breakdown pathway, right? And in the muscle, the reason why muscle is not able to release free glucose because of muscle lacks this enzyme, glucose 6-phosphatase. So this enzyme can work in 2 different pathways, gluconeogenesis and also glycogen breakdown, right? So try to remember these 3 irreversible key reactions in gluconeogenesis, right? The key enzymes. Okay, so this is a little bit more complicated, but kind of overview of the gluconeogenic pathways. Again, gluconeogenic pathway occur in both mitochondria and the cytosol, right? And what you try to remember is that the in the mitochondria, there are actually 2 key reactions, right? In the mitochondria and 2 key enzymes. And in the cytosol, also 2 key reactions, key enzymes, and occurs in the cytosol. This is sort of a typical, gluconeogenic pathways. But I wanted to tell you actually also that gluconeogenic pathway can be different depending on the precursors, right? So, for example, if we our body uses pyruvate or lactate as a precursors, and this will go through the complete gluconeogenic pathway involves the four key reactions, 2 in the mitochondria and 2 in the cytosol, right? And if we use the glutamate, amino acids glutamate as the precursors, and we only involve, the pathway only involves the 3 reactions, 1 in the mitochondria and 2 in the cytosol, right? And lastly, if we use this glycerol as the precursors and only involve the 2 reactions in the cytosol, right? So just try to remember different precursors and uses the different, the gluconeogenic pathways. And amino acid glutamate is also involved with TC cycles for the gluconeogenesis. Any question? Right? Is that clear? Yes.
Are there numbers for the number of reactions in the red box? Are those specific to the number of reactions required?
Oh, here you mean? Yes. Yeah, I just explained, if lactate or pyruvate as the precursors, that goes through the 4 reactions, right? 2 in the, pyruvate, lactate stuff from here. It goes through the 2 reactions in the mitochondria, and last 2 reactions in the cytosol, right? And if glutamate is used as the precursors, and we start from here, glutamate is converted alpha-ketoglutarate, enter TC cycles, and uses some part of the TC cycles and converted to the oxaloacetate, right? And oxaloacetate directly to the PEP, so we'll bypass this pathway, right? If glutamate as the precursors, you only, involve the 3 reactions, 1 in the mitochondria and 2 in the cytosol, right? And glycerol, again, happening in the cytosol, so only goes through the 2 last reactions, right? So shorter, the pathway is shorter compared to the other precursors, right? Is that clear? Any questions? You have a question? Oh, sorry, I thought you was
So, like where you have lactate, there's 4 reactions.
Uh-huh.
It looks like there's a lot more than 4. I don't understand.
No. I mean the 4 key reactions. Sorry.
So
4
everything
key
that's not
reactions.
circled is the exact same
Right.
in glycolysis as it is in
Right.
gluconeogenesis.
Right.
And then the circled ones are what is different because of the irreversible reaction.
Yes.
But I thought there was only 3 irreversible reactions. What's the 4th one?
Oh, here, for example, this pyruvate to oxaloacetate. This is pyruvate carboxylate, right? This is one. And then oxaloacetate to, phosphoenolpyruvate is the second one. So 2 in the mitochondria and other 2 in the cytosol. So, of course, involve lots of other reactions, right? And this, particularly in the glycolytic pathway, this reaction can be reversed. So the same enzyme that catalyze glycolysis can also catalyze the opposite, the reaction in the opposite direction. So you just try to focusing on this 4 again, the key irreversible reactions, right? I don't want to confuse you guys. That doesn't mean it only requires 4 reactions. I mean 4 key reactions. All right. Okay, so I just give you a little more detail, right? The mitochondria, we know the 2 key reactions, right? key reaction key enzymes that you will need to memorize. For example, let's say let's start from lactate, right? Lactate can be converted back to pyruvate and use the same enzyme that in the glycolysis. This is fine, right? So pyruvate and then enter the mitochondria can be converted back to the phosphoenolpyruvate, and this is involved in one reaction, right? So pyruvate first of all converted to oxaloacetate by pyruvate carboxylase. This is the key enzyme, right? Key reaction, and oxaloacetate and then be converted phosphoenolpyruvate and phosphoenolpyruvate carboxyl kinase. This is the second key enzyme. So there are actually two key reaction enzymes that in the mitochondria that involve the gluconeogenesis from either lactate or pyruvate, right? Does that make sense? And phosphoenolpyruvate, once it's made in the mitochondria, then go out of the the mitochondria into the cytosol. So in the cytosol, then PEP, phosphoenolpyruvate can be made. Sorry, here actually, right? And then start from here and go through the opposite direction, that is, to the glycolysis. And this is the last two reactions, the key reactions, right? Irreversible reactions, and you want to remember. So the first is the fructose 1,6-phosphate, which is made eventually from the PEP, right? So reversible reaction, and this molecules can be break down to fructose 6-phosphate by fructose 1,6-phosphatase. This is the one of the key reaction in the cytosol, right? And last reaction would be the and fructose 6-phosphate that I converted glucose 6-phosphate. And glucose 6-phosphate, the phosphate need to be removed from the molecules and release a free glucose. And this reaction is catalyzed by glucose 6-phosphatase. This is another key enzyme that in the cytosol, right? So two key reactions in the cytosol in the gluconeogenic pathways that you wanted to remember, right? So again, if glycerol as the precursors, so they can be first of all phosphorylated to glycerol 3-phosphate, and then to convert it to dihydroxyacetone phosphate, right? And then back to the fructose 1,6-phosphates. And so that's why the glycerol is only involved the two last key reactions in the gluconeogenesis for making glucose. Right? And one thing I just want to point out, although we have not get there yet, but just wanted to remind you that glycogen breakdown, remember I mentioned, right? Pathways can also generate glucose 6-phosphate, and glucose 6-phosphatase is required for free glucose release. So in the liver, liver has the glucose 6-phosphatase, so liver or kidney, right? Both liver, kidney, both have a glucose 6-phosphatase. That's a key enzyme. So that's the reason why in the liver, kidney, they're able to release glucose from glycogen breakdown, but muscle lacks this enzyme, right? So some enzymes, they have like overlapped function. So this is one example. The glucose 6-phosphatase can catalyze this reaction. So this reaction can occur in the gluconeogenesis and also glycogen breakdown pathway, right? So, any questions? Right? Okay, so some kind of details, the last two steps, right? Last two, conversion of fructose 1,6-phosphate to fructose 6-phosphate, right? From this direction, catalyzed by fructose 1,6-phosphatase, and this is again the key reactions, right, in the gluconeogenesis. But in the glycolytic pathway, actually, this fructose 6-phosphate is converted to fructose 6-phosphate, and this is the key enzyme in the glycolysis, right? So they kind of pair, depend on the which pathway and which direction the reaction will go. And again, this is the last reaction. Glucose 6-phosphate can be converted to glucose by glucose 6-phosphatase is in the gluconeogenesis pathways or glycogen breakdown pathway, right? And the opposite direction would be glucose phosphorylate to glucose 6-phosphate by glucokinase, hexokinase, which are mostly active in the glycolytic pathway, right? So, okay, questions? No? Is that clear? Okay, so let's talk about a specific the gluconeogenics pathways cycle. So this sort of kind of big picture, right? The previous couple of slides that we talked about, like in a little more detail, right? Specific reactions in the gluconeogenic pathways, and the key enzyme again, you guys wanted to remember. And first, the specific gluconeogenesis, the Cori cycles. That this it talks about, we know the lactate or alanine are the precursors, right, for gluconeogenesis. But where these molecules comes from, for making glucose, how our body making glucose from these molecules like this. So this is specific pathway called Cori cycles. Basically, is the cycle that muscle and red blood cells are the two main tissue cells that contribute or donate the lactate, right? Remember during the fasting conditions, muscle glucose metabolism will not go complete oxidation, so just stop at the glycolysis. So muscle will release a lot of lactate during the fasting, and red blood cell, of course, is an important place, right, to produce lactate, because again, no mitochondria, so the glucose has to be, go deoxidized at the lactate, right, lactate level. So lactate released from these tissues and cells and transported to the liver. So liver picks up the lactate from the circulation and used for making glucose through the gluconeogenesis, right? So this is the cycles of Cori cycles, and purpose of Cori cycle is to make glucose, right? Gluconeogenesis, again, lactate, start from lactate, and lactate, the muscle and red blood cells are the major contributors, the donors of the lactate for gluconeogenesis in the liver. Alanine. So alanine cycle is the same thing. That the alanine muscle is the major, again, the contributor of alanine for gluconeogenesis in the liver. So alanine can be made from pyruvate, which is mainly from the amino acids metabolism, right? For example, through the transamination, the pyruvate can be converted to the alanine. For some reason, I misspelled this. So alanine then be transported to the liver. In the liver, alanine being deaminated to pyruvate, back to pyruvate. So pyruvate then be used as a precursor, right, for gluconeogenesis in the liver. So liver and then release the glucose to the circulation, try to raise the blood glucose level, right, during the fasting. Any question about these two specific gluconeogenic cycles? Right? Try to remember what is the purpose of this, in which physiologic condition these two cycles occurs, and in the substrate, where the substrates come from, precursors come from, and how they produce and to make glucose. Okay, so I guess I should, let me see. I just try to finish. Okay. Just try to finish this slide, right? And then we have the quiz. So glutamate, I mentioned, right, it's also can be used as a precursors, but only involve the three key reactions in the gluconeogenic pathways, right? Glutamate, first of all, be converted to alpha-ketoglutarate, and then enter the TCA cycles. So TCA cycle is also involved in the gluconeogenesis through, from the glutamate, right? So what you try to remember here is if we use the glutamate as the precursors, first of all, only involve the three key reactions, right? And one in the mitochondria and two in the cytosol. And secondly, TCA cycle is part of a gluconeogenic pathway if we use the glutamate as a precursors, right? Does that make sense? And lastly, this gluconeogenesis from glutamate actually bypass the pyruvate kinase key reaction, this reaction, right? Okay, any questions so far? Right? Okay, now it's quiz time.
Me too, and I'm not